What's up, everyone? It's Mr. Maramba coming at you with more science. To continue our review for our final, we will move into a lesson reviewing experiments. Specifically, we'll be going over 3 different parts. Our first part is identifying the parts of an experiment. The second is reading and interpreting data tables and graphs. The third, being able to predict experimental outcomes based on those data tables and graphs we're provided. So let's go ahead and first identify the different parts of an experiment. All experiments have an independent variable, a dependent variable, and at least one control variable. Every single experiment also has something that's called an experimental group. And lastly, in controlled experiments, you also have the addition and inclusion of something called the control group. So the independent variable is the thing that changes on purpose. There may be more than one, but in order to identify it, we need to see what the scientist, what the experimenter is changing on purpose in their experiment. Here, if we look at the data table, the thing that is changing on purpose is the number of lights. So we have the number of lights changing between each group that's being experimented on. And to jog our memory, this is an experiment on seeing how different plants, different seeds grow, which is why we have different pots to experiment on. We also have an independent variable that is not seen here. This is the number of days. The experimenter here is choosing to perform the experiment over the course of 50 days. So the independent variable, in summary, is what changes on purpose or what does the effect. And in our case, in this experiment, it is the number of lights and time in terms of days. The dependent variable, on the other hand, is what's being measured or what is affected, what is dependent on the independent variable. So we see at the bottom here that our dependent variables, we can have multiple, are both height and mass at the end of 50 days. So height and mass depends on time, so 50 days, and it also depends on the number of lights there is. Our control variables is everything else, or what stays the same throughout each group in the experiment. So if we look at groups A through G, we will see that what stays the same is the type of seed in each pot, the water per day in each pot, the type of soil, which is compost and fertilizers, and among other things as well. Experimental groups are the groups that are being tested. And this is different from the control group because the control group is the group that is not affected by the independent variable. And it is meant, like the control variables, to isolate that independent variable, meaning it's trying to make sure that nothing else is affecting the dependent variables. It's only the independent variable, it's only the amount of sunlight here that is affecting height and mass. It's not anything else. So these are the different parts of an experiment. Make sure to keep those in mind and memorize. Let's go ahead and go over reading data tables and graphs. On the left, I have a data table. On the right, I have a graph. You already are familiar with data tables and are able to see pot A, pot B, and pot C. Pot A has 1 light, pot B has 2 lights, pot C has 3 lights, and the rest of the other variables are control variables. On the right, we have, that's going left and right, the thing called the x-axis. In this case, it is showing the independent variable, and it will always show the independent variable. It includes the different amounts of light in different colors. So pot A is red, pot B is green, pot C is blue. But more importantly, it shows another independent variable that is changing on purpose, and that is time. We can see that a graph is appropriately titled height versus time, which includes, guess what, the other independent variable, the different amounts of sunlight. 1 light, 2 light, and 3 light, and etc. On the y-axis, the thing that goes up and down, you always have your dependent variable. So what is being measured? This, in this case, is the height. If I go over to mass, mass is also on the y-axis because the y-axis always has a dependent variable, the x-axis always has the independent. Lastly, let's go over our third part, which is reviewing how to predict the experimental outcomes or what could happen in an experiment. So here we're really analyzing data and seeking patterns. So we're not only looking at the evidence, what our data shows, we're also interpreting it in order to come up with an appropriate claim, conclusion, or stance or argument. Here I have a last data point set missing. G is blank. But based on this pattern, what would be the height of pod G and what would be the mass of pod G? If we look at the pattern, the height goes 0, 5, 6, 7, 8, 9, and we have 5 being 4 grams, 6 being 6 grams, 7 being 8 grams, 8 being 10. We could tell that pod G would probably be 10 centimeters in height and 14 grams in mass, considering the pattern before in the other pots. So because we see this pattern of addition in the other groups, groups A through F, we can conclude that pot G has a height of 10 centimeters and a mass of 14 grams. That is how we use evidence and interpret evidence in order to come to a claim in which we will support with that evidence and interpretation. Because height is going up by 1 from groups B to all the way F, and mass is going up by 2 between each group from groups B through F, then I can conclude that group G would have a height of 10 centimeters and a mass of 14 grams. That concludes our lesson on experiments. There are a lot of definitions and terms you must remember, so vocabulary is especially important. For our exit ticket and classwork today, you will complete the questions on the study guide, which are all open response. Be sure to review this video if you have any questions and reach out if you need me. And I hope to see you all soon. Thank you very much.